Good morning, everyone, and welcome to the Tibor T. Polgar Fellowships Final Reports presentations. I'm Helena Andreyko. I'm the administrator of the Hudson River Foundation, and on behalf of the foundation, I want to say how happy we are that you all could join us this morning. So each year, the foundation awards up to 8 Polgar Fellowships, and today we'll hear about the research of our 2020 fellows. I'm joined on the screen by a group of very distinguished people, and we'll begin the program by introducing ourselves, and we'll start with the Fellowship Committee. So, Sarah?
Hi, I'm Sarah Fernald. I'm the research coordinator for the New York State DEC Hudson River National Estuarine Research Reserve.
Thank you. Dave?
Dave Yazzo with Glenford Environmental Science. I've been involved with the foundation and the Harbor Estuary program for many years on a variety of programs and initiatives. Notably, since 2008, I've been the co-administrator of the Polgar Fellowship Program, along with Sarah and Helena.
Thank you. And of course, our 2020 fellows. We'll start with Sarah.
Hi, I'm Sarah. I'm a Columbia master's student, and I work with Wildlife Conservation Society's Ocean Giants program.
Okay. And Emma?
Hi, my name is Emma Garrison. I'm a student at the School of Earth and Environmental Sciences at Queens College. I'm working with Dr. O'Mullen, and I'm a Polgar Fellow.
Yay. Aslan?
Hi, I'm Aslan McBull. I'm also at the School of Earth and Environmental Science at Queens College. I'm also in Dr. O'Mullen's lab. I'm also a Polgar Fellow.
Great. And Eva?
Good morning, I'm Eva Hasegawa. I'm a second-year master's student at the NYU School of Medicine, and I'm in Dr. Wergen's lab, and I'm also a Polgar Fellow.
And we're also joined by our staff member, Sam.
Hi, I'm Sam Gordon. I'm the program assistant with the Hudson River Foundation.
And Sam's going to be helping us with any technical issues we have, and he's also our host. So our agenda today will begin with an overview of the Polgar program and the Hudson River Foundation presented by Dr. Yazzo, followed by an introduction to the New York State DEC Hudson River National Estuarine Research Reserve, the co-sponsor of the Polgar Fellowship, presented by Sarah Fernald, and then we'll hear from our fellows. So each fellow is allotted a half an hour of time. At the end of each fellow's presentation, we'll open the floor to questions, which you are free to put in your chat, which you find at the bottom of your screen. We'll try to get to as many questions as we can while staying within our schedule. And if we have to move on because we've run out of time, we'll try to circle back at the end to catch up. And with that, I'm going to turn the program over to Dr. Yazzo.
Thank you, Helena. I'm going to go ahead and share my screen and hope for the best. If it doesn't work, we have Sam standing by. Can we see the presentation?
Yes.
Okay, good. Full screen it on my end. Okay, so we're going to do this a little bit different this time. Previously, we've done this in person at the Carey Institute with a much smaller audience who may be a little more familiar with the foundation, but we have a much broader, diverse audience today via the virtual platform. So I'm going to give a little bit of historical background on how the foundation, how the Polgar Fellowship Program came to be. It's 3 decades on the Hudson River Estuary because this is the 36th year of the program. You could say the foundation had its origins way back to the Clean Water Act, 1972. Section 316B specifically required cooling water intake structures at power plants and other facilities which withdraw cooling water from natural water bodies to use the best technology available for minimizing adverse environmental impact. At the time, the EPA and New York DC decided that cooling towers were the best available technology for minimizing adverse environmental impact at 3 Hudson River cooling water intakes in particular. However, the utilities said, no, they do not. And began a lengthy, expensive, and non-productive history of litigation and settlement from 1975 to 1980. Ultimately, there was a settlement, and the settlement agreement resulted in the implementation of a series of interim mitigation measures to be protective of fish and aquatic resources at those power plants, but not cooling towers. Operational and technological upgrades, including barrier systems and a variety of other technologies that we don't have time to go into today, also led to the formation of a program of environmental studies that ran from 1974 to 2018, the Hudson River Biological Monitoring Program, which resulted in a tremendous long-term data set on the Hudson River. It's been used by many, many researchers and over the years. It's a really unique data set, and also resulted in the formation of the Hudson River Foundation. So the foundation was established in 1981 with an initial endowment of $12 million. This was then invested and turned into a lot more money. And that allows the foundation, primarily through its Hudson River Fund, but also through a number of other grant programs historically, to have funded over 800 grants, research grants, totaling in excess of $40 million over its history. But in addition to just funding research and public policy and other grant programming, the foundation does quite a bit more. They sponsor the Harbor Estuary Program since 1987. They have sponsored the Oyster Restoration Research Program in more recent years, the Contaminant Assessment Reduction Program. They're an essential partner in the Hudson Raritan Estuary Comprehensive Restoration Plan, plus a variety of other research, educational, public involvement type programs. Really don't have a lot of time to go into those now, but if you go to the Hudson River Foundation website, you can find information on all of these programs, which I recommend that you do if you're new to the foundation. But the program we're going to talk about today is the Tibor T. Polgar Fellowship Program. The program was started in 1985. Initially, research was limited to the 4 component sites of the Hudson River National Estuarine Research Reserve, which Sarah is going to talk about in more detail in a few minutes. So Piermont, Iona Island, the Tivoli Bays, and Stockport Flats. But since 1988, the program has expanded to include research within the entire estuary. However, many of them do still take place in the research reserve because the reserve provides a lot of support, research ideas, collaboration opportunities for students. So we still do see quite a bit of emphasis in the reserve sites. As I mentioned, the program has been going on for 36 years. 2020 is the 36th cohort, and we have funded 284 fellows to date with over $1.2 million in support. If you're not familiar with who Tibor Polgar was, he had a very interesting life history for sure. He was born in 1944 on a train to Auschwitz, and his mother hid him under the floorboards in the sleeping quarters of the camp, where she was a seamstress. He, his mother, and one uncle out of his entire extended family survived the Holocaust, and they moved back to their native Hungary. However, in 1956, they were forced to flee Hungary because of the Hungarian Revolution, and he was in one of the very last groups to immigrate to the United States through Ellis Island. I did not know Tibor, but I do have some colleagues who went to graduate school with him, who worked with him. He became a very accomplished fisheries population biologist. He studied at the University of Rhode Island. He went on to work in the Hudson and other mid-Atlantic estuaries, focus on anadromous fish. And he worked at the Martin Marietta Environmental Corporation, which then became Versar. Actually, if you go to Versar's headquarters in Columbia, Maryland, they have a very nice memorial in their lobby to Tibor. Apparently, he was also extremely talented musician. He was an accomplished violinist and played with the New York Philharmonic while still a teenager. He was the first chairman of the Hudson River Science Panel in the early years of the foundation, but he died at the age of 41. And so he, shortly after his death, the program was named in his honor. The basics of the program are that students, they can be undergraduate, master's, PhD, they submit research proposals. These proposals, it's a competitive process. The proposals are reviewed internally by our committee, other staff members at the foundation, and at least 2 external reviewers. So it is a competitive process. The duration of the research is typically around 3 months. It's considered a summer program, but we recognize that many of these projects are part of a longer term effort, a thesis or a dissertation, for example, especially for the graduate students. So we recognize that these are a component of a larger research effort or program. Advisors play a critical role at the host institution. They supervise all aspects of the research. They edit the final reports and they participate in orientation meetings, which are typically held in the late spring, and then a final presentation meeting, which is typically held at the end of the summer. Although, of course, this year with COVID, we've had to change things up. We had an orientation meeting yesterday, and now we're having our final presentations today in a virtual format. The stipends are $5,000 to the fellows. The advisors get a $500 stipend as well, and there's a modest research budget, about $1,000 to $1,500 to support the research. And then the program for the students culminates in the event that we're having today. Again, this is the first time we've done it in a virtual format. Typically, we do this at the Carey Institute of Ecosystem Studies. They have been excellent hosts for the final presentation gathering for many, many years. I just want to, shout out to Stuart Finley. He has every year been a terrific host for this event. We really enjoy working with him. And, I don't know if it's going to happen in 2021, but hopefully the following year, maybe we can get back to the Carey Institute, because it's a great place to do this. And then at the end, the students have to write a report, and they are rigorously edited and reviewed by our panel, and they are published in a bound volume and a digital version by the Hudson River Foundation, and they are fairly widely distributed among the academic research community in the Hudson River Valley and beyond. And of course, we encourage the fellows to publish their results in the peer-reviewed literature, and many, many of them have done so. And finally, I just want to mention one other program, if you're not familiar with it. If you're a graduate student moving on for a year of support at the master's or doctoral level, there's the Mark Bain Graduate Fellowship. Hudson River Focus is essential for this. It is very competitive. And Mark, if you don't know who Mark was a sturgeon biologist at Cornell University. For many years, he worked with the foundation as a very close advisor, especially during the foundation's involvement in the comprehensive restoration plan for the Hudson River Estuary. And so they named the graduate fellowship program after Mark. And again, if you're interested in more information, how to apply for the Bain Fellowship or the Polgar Fellowship, I encourage you to go onto the Hudson River Foundation's website and get all that information. And now I'd like to turn it over to Sarah Fernald, and she's going to talk about the Hudson River National Estuarine Research Reserve.
Thank you, Dave. Let me try to share my screen. All right, can you see my presentation? And it's in full screen mode? Great. So hi, I'm Sarah Fernald. I'm the research coordinator for the New York State Department of Environmental Conservation Hudson River National Estuarine Research Reserve. We go by the shorter acronym HRNR quite often, and I manage the research and monitoring program. HRNR is one of 29 research reserves across the country. Most coastal states have at least one. Connecticut is coming on board shortly to bring us up to 30 research reserves. And all of the reserves share a mission to promote stewardship of the nation's estuaries through science and education using a system of protected areas. We are administered through the National Oceanic and Atmospheric Administration and funded through a congressional appropriation. And these are administered through a federal-state partnership. Our state partner happens to be the New York State Department of Environmental Conservation. We were designated in 1982 and consist of 5,000 acres of protected wetland across 4 component sites. And those are Piermont Marsh, which is our southernmost site, located just south of the Mario Cuomo Bridge, formerly the Tappan Zee Bridge. Iona Island is located in Bear Mountain State Park. Tivoli Bays is adjacent to the Bard College campus, and Stockport Flats is just north of the city of Hudson. Pictured there is our headquarters at the Norrie Point Environmental Center, which is between Poughkeepsie and Tivoli. Within our reserve program, we have several sub-programs which we call sectors. Like I said, I lead the research and monitoring sector. We also have an education and outreach program, folks that focus on stewardship and restoration, and a coastal training program which translates the science for end users and stakeholders so that the information that we're gathering is readily usable, and our other goal is that all of our data is publicly accessible for students such as the Polgar Fellows to make use of. So most of our data is collected within what the NEARS funds as the System-Wide Monitoring Program, or SWAMP. We do abiotic monitoring where we look at nutrients, collect meteorological data from weather stations, and monitor water quality with YSI sondes. And we also look at biological monitoring, focusing on submerged aquatic vegetation and emergent marsh habitat, and collecting data on the spatial distribution and vegetation characteristics within those habitats. So our abiotic monitoring has been ongoing since 1991 with our nutrient collection from all of our component sites. Since 1995, we've done more intense continuous water quality monitoring, for most of that time focused in the Tivoli Bays component site. We were headquartered at Bard College for quite some time before moving to our Norrie Point facility. So when we made that move in 2007, we started monitoring water quality there as well. And just recently, one of our pandemic projects was adding some new stations at our Stockport Flats component site at Ferry Landing, a new dock that the stewardship program constructed there, and another water quality station at Iona Island on a dock in Bear Mountain State Park. We also have 2 meteorological stations, one at Tivoli Bays and one at our offices at Norrie Point. So like I said, all of this data is publicly accessible. We highly encourage researchers and students to make use of it as baseline information as they're planning and implementing their projects. It is accessible 2 ways, through the NEARS Centralized Data Management Office website. And we also connect with the local network, the Hudson River Environmental Conditions Observing System. And all of our data streams live to the site. It's collected every 15 minutes and runs continuously, except we do pull a few of our stations in the winter due to ice formation. Within the New York State DEC, we're housed within the Hudson River Estuary Program's Habitat Unit in Region 3, where we look at habitat protection of submerged aquatic vegetation and tidal wetlands. So to monitor these habitats, we've been mapping them. We have a series of 6 submerged aquatic vegetation maps and are working on our second interpretation of tidal wetlands from aerial photographs. And these are the full stretch of the tidal Hudson River estuary from Hastings-on-Hudson to the Troy Dam. And again, all of this information is publicly available on the New York State GIS Clearinghouse. The address is there below. And a great resource again for researchers just to understand the vegetation distribution at their proposed study sites. So we also conduct some research on this habitat, mostly looking at marsh resilience to sea level rise. So as we collect the information on what species occur in what locations, most of the tidal marsh vegetation is really dependent on the elevation to determine which species thrive best there. We also know that over time, the elevation is changing as more sediment is accreting or eroding in certain places. So we're monitoring marsh elevation change over time with the surface elevation tables, and also looking at the sediment supply. We're fortunate in the Hudson, it's fairly sediment rich and is accreting a decent pace, but to make sure it's resilient, we're also tracking the rate of sea level rise to ensure that the marshes are accreting at a pace that's keeping up with the rate of sea level rise for our area. And tidal range is also another key component of marsh resilience. The Hudson has a 3 to 5 foot tidal range, and the bigger the tidal range, the more resilient. Because if you would experience a foot of sea level rise, say over the next century, because it's already a 3 to 5 foot range, it's not felt as much as an area with a lower tidal range. So the reserve did construct a water level station that's connected through NOAA's tide monitoring network. It's located at Turkey Point near our Tivoli Bays component site so that we can get measures of sea level rise for the upper Hudson related to our component site there in Tivoli Bays. And again, all this information is publicly available and ready for students to make use of, as well as other researchers. So I'm going to conclude it there. Thank you very much. If anyone has any questions about the reserve, feel free to contact me. I'm not going to take specific questions about the reserve at this time. We want to focus on the wonderful work that our students have done today. So as they give their presentations, if you have any comments, support, and especially encouragement for how the students can make their analyses in final papers even more robust, we welcome any comments to be typed into the chat box, and I'll help the students field those questions as we go. So with that, I'm going to stop sharing my screen and introduce our first speaker, which will be Sarah Trabue. She is from Columbia University, and she will she has 2 advisors, Howard Rosenbaum and Melinda Rechtal. She'll be speaking on photo identification and skin lesions and prevalence in bottlenose dolphins in the New York, New Jersey harbor estuary. So take it away, Sarah.
Thank you. Can you hear me?
Yes, and I can see your presentation.
Perfect. All right, so thank you for that introduction. And before I dive in today, I'm just going to give a brief overview for how this presentation will flow. So first, I'm going to go over the rationale for this project, why it's important and interesting and relevant. Then I'm going to go over the methods for the 5 sections that made up this project. So that included building a photo ID catalog, categorizing skin lesions, looking at temporal and spatial variation in lesions, as well as investigating potential environmental influences on lesion development. I'll then go over the results for each of those sections and discuss my findings. So just to orient you, this project was conducted in the New York, New Jersey harbor estuary and the surrounding waters. So that's right about there. And one little known fact about this, this area is that for part of the year, it's home to bottlenose dolphins. So bottlenose dolphins that are found off the coast of New York and New Jersey belong to the Western North Atlantic migratory coastal stock. So this stock spends cold water months in the coastal waters of North Carolina, as indicated by this blue shaded region down here. And when it's in that southernmost part of its range, it overlaps with other stocks, both spatially and temporally. During the warm water months, this stock migrates northwards and spends its time off the coast from Assateague, Virginia, all the way up to a little bit above where this map cuts off the waters of Long Island, New York. So bottlenose dolphins can be found off the coast of New York and New Jersey from about April to October every year. And this seasonal migration is likely driven by fluctuations of water temperature as well as feeding habits. So it's exciting unto itself that there's bottlenose dolphins in the waters off of New York and New Jersey. But even more good news is that recently, these dolphins have been seen both more frequently and for an extended period of time in this area, suggesting that recent efforts to restore the habitat quality of this area have made the environment more viable for marine mammal use. However, the New York, New Jersey harbor estuary and surrounding waters are still a stressed out system with many anthropogenic pressures. For example, the harbor estuary is home to the largest port on the eastern seaboard, so there's lots of vessel traffic. So we viewed 2020 as a unique opportunity to monitor this population of bottlenose dolphins during a period of reduced anthropogenic pressure due to the COVID-19 global pandemic. And this research contributes to a larger body of work that's currently being done to look at which marine mammal species are found in the harbor estuary and surrounding waters and how and what they're doing there. So my master's thesis focuses on bottlenose dolphins, and there's other components of my thesis that look at the foraging and social activity of these dolphins in this area using bioacoustics. But the purpose of this project was to gain a better understanding of population health in these animals. And in order to do that, I looked at skin lesions. So skin lesions are damaged skin tissue that can be caused by either injury or disease. If it's disease, it can be viral, fungal, or bacterial in origin. And when you see an individual that has skin lesions present, that indicates that this individual is either diseased or of diminished health, for example, has a depressed immune system. The presence of skin lesions can also indicate that there are stressors in the environment, and these could be natural stressors such as sea surface temperature or salinity, as well as anthropogenic stressors such as chemical pollutants. So here along the bottom, these photographs are from previous studies where skin lesions have been used to infer and monitor population health in other populations of other species. So on the bottom left, you can see that skin lesions were looked at in critically endangered Indo-Pacific humpback dolphins. In the middle, they were looked at in critically endangered North Atlantic right whales. And on the right, they were looked at in humpback whales. So skin lesions have been documented in bottlenose dolphins for about 2 decades now. But there's still little known about patterns of development and distribution across these populations. And one method that's been used more broadly to monitor marine mammal populations in the past has been the use of a method called photo identification, or photo ID. And the basic principle of photo ID is that you can take a photograph of an individual marine mammal, in this case a bottlenose dolphin, from the side view, and you can use distinctive notches in its dorsal fin in order to identify that individual based on the photograph alone, hence photo identification or photo ID. So photo ID has been co-opted for skin lesion analysis because skin lesions tend to show these macroscopic qualities such as size, shape, color, and apparent texture, and these can be described, categorized, and potentially used to infer the presence of underlying diseases. So some benefits of photo ID include that it's cost-effective, it's simple, it's non-invasive, so you don't stress the animals out too much. And again, because photo ID has been used historically to monitor marine mammal populations, it's possible to go back into those archival data sets, those photographs from the past, and screen them for lesions and get an idea of what population health might have been like in the past. Some limitations of photo ID include being limited by the quality of the photo, for example, if there's poor lighting. You're also limited by what parts of the dolphin's body are visible to you. So typically that's going to be the dorsal surface of the dolphin's body, even though lesions can be present anywhere on the body. So any estimates that you get of skin lesion prevalence are going to be minimum estimates. Photo ID is also subject to some level of inter-observer bias, and it also does not give a conclusive etiology or cause for the skin lesions. However, despite these limitations, using photo ID for skin lesion analysis provides valuable information for inferring population health, and that can provide a baseline for comparison across both space and time. So the overarching aim of this project was to gain a better understanding of population health in these dolphins by using photo ID to investigate patterns of skin lesion prevalence. And we had several questions pertaining to this overarching aim. The first of which was, do dolphins in this area exhibit stable seasonal patterns? And this is an important population characteristic to establish because you want to make sure that you're looking at a representative sample from the same population when you're drawing conclusions about disease prevalence over time, as opposed to looking at samples from different populations that might be exposed to different stressors, because that would confound the data. We were also interested in the number and type of lesions observed on these dolphins, whether lesion prevalence was stable over time or across space, and what might be contributing to lesion development in the waters off of New York and New Jersey. Okay, so in order to investigate these questions, boat-based surveys were conducted by the Wildlife Conservation Society's Ocean Giants team during the months of May to October and during the years of 2017 to 2020. So on this map to your right, you can see all of our bottlenose dolphin sightings. And on the left, you can see more specifically in the harbor estuary. And each of these data points represents a time where bottlenose dolphins groups were observed. They're not individual dolphins. And you can see that they're colored by the year that they were sighted. So during these boat-based surveys, photographs of bottlenose dolphins were collected. In order to build a photo ID catalog of unique individuals, each of those photographs that was collected during the surveys were screened for distinct individuals. Distinct individuals, once identified, were then compared to all other distinct individuals. And they were either a match, and that's constituted a recite, or they were a new individual, in which case they were added to the catalog and given a unique ID. Then before we ran the lesion analysis, we removed any catalog photos that had poor lighting where you couldn't see the surface of the skin. We removed catalog photos with only the dorsal visible, and we also removed within year resites because it's still unclear how long lesions can persist on the surface of the skin. So if a dolphin was seen multiple times within 1 year, only the first sighting of that year was used in the lesion analysis. Once these ineligible photos were removed, we noted whether lesions were present on an individual and the type of lesions that were present on an individual using the 17 categories put forth by Toms et al. 2020, plus an additional other category. So you can see that on the table down to your left. And I want to quickly draw your attention to these two categories here, pathogenic rake marks and non-pathogenic rake marks. So rake marks are injuries that can be caused when there's aggressive contact between the teeth of one individual and the surface of the skin of another individual. And it tends to form these long, thin parallel lines that can be either hyper or hypopigmented. So you can see an example of non-pathogenic rake marks in this photograph to your right. And in the past, when there's in studies that have looked at skin lesion prevalence in other populations and other species, rake mark associated lesions or RMA lesions have not been included in the skin lesion analysis. So going forward in this presentation, when I refer to skin lesions or lesion prevalence, I'm talking about these other categories, not RMA lesions. We also looked at temporal variation, and the basic equation we used to look at prevalence was the number of individuals with at least one lesion for whatever time period was of interest, divided by the total number of individuals screened for lesions during that time period of interest. So we did that across the entire catalog in order to get overall prevalence. We did it by year and we did it by month. We were also interested in spatial variation because, again, the population that's found in the New York, New Jersey harbor estuary and surrounding waters belong to a migratory population. And when they're down in the southernmost part of their range off the coast of North Carolina, they overlap with other stocks, both spatially and temporally. So we were interested in whether spatial variation was stable over time, or sorry, if prevalence is stable across space. So we compared our overall estimate for the New York, New Jersey harbor estuary to other populations in North Carolina, South Carolina, Georgia, and Florida. We were also interested in environmental influences on lesion development. So in order to investigate this further, we broke up the season of residency into 22 weeks, starting May 31st and ending October 31st. We calculated weekly lesion prevalence, average salinity, and average sea surface temperature, and put those into a generalized linear model. And this is what we found. In the final photo ID catalog, there was 221 unique individuals, 7% of which were recited and 3% of which were recited within the year. Once all ineligible photographs were removed for the lesion analysis, there's 203 photographs remaining. And here we have at the bottom is an example of a recite that we had. This is individual 71. You can see on the bottom left that it was observed during the fall of 2018. And then on the bottom right, it was observed again the following spring in May 2019, sporting a brand new lesion to the left of its dorsal there. When we categorize the lesions based on the 17 categories put forth by Toms et al., you can see the spread here on this graph to your left with the categories along the x-axis and the number of individuals with each type of lesion along the y-axis. And we found the 5 most common lesion types were cloudy white spots, white amorphous lesions, mottled lesions, tattoo lesions, and dark fringe lesions. So those first 2 have been previously associated with herpes virus, and those last 2 have been previously associated with pox virus. We were also interested in the number of lesion types seen on each individual at a given time. And so the number of lesion types ranged from 0 lesion types seen on an individual to as many as 4 different lesion types seen on an individual at a given time, with the mean being 0.57. Now, if you look at this bar graph to your left, you'll see along the x-axis, we have the number of lesion types, and along the y-axis, the percent of individuals with that number of lesion types. And it essentially breaks down like this. About 60% of our individuals had no lesions, about 30% had 1 lesion, and about 10% had 2 or more lesion types. We first calculated overall prevalence when we were looking at temporal variation, and we found that across the entire catalog, about 41% of our individuals had at least 1 lesion. When we broke it down by year, we found that 2017 was the year with the highest prevalence, with about 50%, 1/2 of individuals having at least 1 lesion. The year with the lowest prevalence was the following year in 2018, where only about 30% of individuals had at least one lesion. And I want to draw your attention to 2020. During this period of reduced anthropogenic pressure, we see a slightly lower prevalence, but still 10% higher than it was 2 years previously in 2018. When we looked at temporal variation by month, we only had one sighting from May, so we excluded that sighting from the analysis in order to not skew our data. But we found from June to October that the highest prevalence was in the earliest month of June, and then it decreased over the summer and fall months. And when we looked at spatial variation, we found that prevalence estimates for the New York, New Jersey harbor estuary and surrounding waters were within the range of other sites along the East Coast, but they were relatively higher than most sites with the exception of Florida, where the prevalence was roughly equivalent between sites. We also investigated the potential environmental influences on lesion development, and from our generalized linear model, it was suggested that salinity was the only significant variable. So if you look at this graph on your right, you'll see that as average weekly salinity increases along the x-axis, skin lesion prevalence decreases along the y-axis. So this suggests that in fresher waters, you would expect to find higher prevalence of skin lesions than you would in saltier waters. And this is consistent with previous findings that have found that when there is reduced salinity, it's been significantly correlated with specifically the development of poxvirus associated lesions. So this is what we think about what we found. From the photo ID catalog, the timing of resites suggests that there are indeed stable seasonal patterns for dolphin presence in the New York, New Jersey harbor estuary and surrounding waters. And again, this is an important population characteristic to establish when monitoring health of this population. When we categorized the lesions, we found that the most common lesion types were associated with viral infections, namely herpes virus and poxvirus. Herpes virus has a low mortality rate in bottlenose dolphins, but has been occasionally associated with fatal infections of the nervous system. And herpes virus is hypothesized to spread through the sexual contact of free-ranging bottlenose dolphins. Poxvirus also has been associated with a low mortality rate, but indicates diminished health and has been associated with calf mortality when there are multiple concurrent stressors. And potential causes of poxvirus include reduced salinity and the presence of chemical pollutants in the environment. We also found that 11% of our individuals had 2 or more types of lesions, suggesting there is a potential for interactions between multiple kinds of underlying disease. When we looked at spatial and temporal prevalence, we found that prevalence was not stable across time or space. When we compared prevalence in the New York, New Jersey harbor estuary to other sites along the Atlantic, we found it had a relatively low prevalence. And also when you compare it to other sites globally for bottlenose dolphins, it has a relatively low prevalence compared to those as well. Interestingly, 2020 did not have the lowest prevalence. In fact, it was 10% higher than the year that actually had the lowest prevalence, which was 2018. So even though there was reduced anthropogenic pressure, we did not see a substantial decrease in prevalence. And when the apparent negative monthly trend that we observed is consistent with previous studies that have showed that prevalence is highest in the springtime and then decreases over the summer and fall months. And we also, with regard to environmental influences, poxvirus-related lesions have been commonly associated with reduced salinity, while herpes virus-related lesions have not. So this suggests that using lesions as an indicator for stressors in the environment may be different based on the type of lesions that you're seeing. For example, if in our population we saw a lot of herpes virus associated lesions, we would not expect, that would not signal that there are stressors in the environment, because herpes virus is hypothesized to spread through social factors. On the other hand, if we see a lot of poxvirus associated lesions in our dolphins, that would suggest that there might be further research needed to see if there are stressors in the environment, because poxvirus lesions have been associated with environmental drivers. So the main takeaways from this project include that lesion prevalence is relatively low in the northern range of the Western North Atlantic migratory coastal stock. And this is consistent with the hypothesis that with recent efforts to restore and improve the habitat quality of the New York, New Jersey harbor estuary and surrounding waters, that it is making that environment more viable for use from marine mammals. So this research is a critical first step for understanding population health in these animals, but more information is needed. For example, we need more information on the causes of lesion development, whether that's anthropogenic or environmental causes, as well as the effect of multiple concurrent stressors on lesion development. And more information is needed on the effects of these lesions and their underlying diseases on survival and fitness. Furthermore, there are future developments that are slotted to occur in this region, including offshore wind. And these developments are cause for concern for this population of bottlenose dolphins. So this work lays a groundwork for how to monitor these populations as these developments roll out in order to see if they're negatively impacted. So in the future, photo ID efforts should continue in order to expand the catalog and also to get a larger sample size on resite data. Because if we have a larger sample size for resites, we can start to look at longitudinal patterns across individuals as well as longitudinal patterns for the population as a whole. This data could be supplemented by other methods of assessing dolphin health, for example, collecting samples from stranded dolphins. Future studies should investigate the influence of anthropogenic stressors, such as pollutants, and also investigate the cumulative and interactive effects of multiple stressors on lesion development. And this is the groundwork that I'm trying to lay with my master's thesis by looking at the foraging and social activity of dolphins and how they might be affected by anthropogenic and environmental drivers, and looking at population health. So in the future, it can provide a holistic view of these bottlenose dolphins and how and what they're doing in this area. So I wanted to end with a photograph of some happier dolphins because this presentation had a lot of sad dolphins throughout it. I want to say big thank you to the folks over at Hudson River Foundation for providing support for this project, as well as to my advisors at Wildlife Conservation Society, that's Dr. Howard Rosenbaum and Dr. Mindy Reckdahl. And I want to say thank you also to the Ocean Giants team, the whole Ocean Giants team at Wildlife Conservation Society for providing so much support. And thanks to everyone for coming today and listening to me talk about lesions. And I'm happy to take any questions.
Thank you so much, Sarah. We do have a few questions and comments in the chat that I'll read out to you. So from Georgina Coleman, she says, Sarah, cool study. There was a presentation at a recent student conference on conservation science at the AMNH in 2020 using a similar methodology with humpback whales. In case of interest, it was entitled Using Drones to Assess the Health and Endangerment of Arabian Sea Humpback Whales by Wong Kant. So just a helpful reference for you there, and I could make sure you get that off the saved chat.
Thank you.
But we also have a question from Dave Bushek. He asks, do you think the level of freshwater inflow is a factor in the spread of pox virus? And do dolphins respond behaviorally by moving to more saline waters? And also adds, nice presentation.
Thank you. So in previous studies, yeah, it's been shown that reduced salinity, so fresher waters, are correlated with pox virus lesions. So that would be worth studying further in the New York, New Jersey Harbor Estuary, whether the flow of fresh water from the Hudson River might be affecting that. In terms of behavioral reactions, I don't have that information, but I could hypothesize that it would depend on kind of food availability.
Great.
Yeah. Thank you for the question.
Yeah. Also, Andy Bernick asks, does WCS or other entity maintain the database of marine mammal sightings in the New York, New Jersey Harbor Estuary, particularly one that covers the East River from the Upper Harbor to Long Island Sound?
So that might be a question better directed towards my advisor, but I know that there are multiple organizations that collect sightings data for marine mammals in this area.
I'd be happy to speak to that, Sarah, if you would like me to.
Yeah, go ahead.
Thank you, Andy, for the question. Yeah, it's a really important question, particularly as we're seeing this resurgence of marine mammals in the New York, New Jersey Harbor Estuary and throughout the New York Bight. And there are a number of entities that are working in the area, and we've worked to establish on one particular species like humpback dolphins a collaborative effort to maintain an online photographic database, for example, in an online platform, and that involves various entities working in and around the entire New York Bight, the Harbor Estuary, and even into the Sound. So that includes Gotham Whale, Cressley, Stony Brook, and the Atlantic Marine Conservation Society as well, along with the Wildlife Conservation Society. And so we've actually had some great discussions at the Hudson River Foundation about the importance of these types of online catalogs and the need for them. And we've kind of got a pilot up, and we're hoping to continue that effort and even have hopefully a public facing side as well.
Thanks, Howard.
Great. I don't see any other questions in the chat right now, but I did have one question myself. You had mentioned that you would have expected 2020 to have shown some signs of the population being a little healthier with the reduced impacts of human activity. But do you think that might just be delayed, and looking into 2021, those impacts from 2020 might be seen later as the population's health improves?
Yeah, I think that's a fantastic question. And I think that's part of why these efforts should continue, because there might be a delay. There's just still pretty little known about how long these lesions persist, or those kind of data is just not there yet. But it would be good to continue it and see if you see this lag.
And Sarah, might I hop in just for a second here too to add a point about Sarah's work here and the importance. I mean, this is a really nice study and designed to look at what one can see when in terms of an anatomical aspect, a phenotypic aspect of potential issues or impacts related to dolphins. Some of the work that Sarah alluded to, which forms the basis of her master's work, is more cryptic, more subtle, which is looking at the impacts of the acoustics in terms of anthropogenic noise on foraging buzzes, and she alluded to that. So it's the duality nature in terms of understanding this whole system, which is really nice in terms of the impacts that are occurring to these animals in the New York, New Jersey Harbor Estuary, and that's work that we've been doing at the Wildlife Conservation Society. It's been supported by the Hudson River Foundation, and Sarah Trabu is doing a nice effort as part of her master's work, as a complement to the Polgar work that she presented. So, and just one thing, and I'm probably going to embarrass Sarah Trabu here a little bit, just for one moment, was that Sarah shared with us, Mindy and myself, that this has sort of been a lifelong dream for her, and through her family dug up a blog that I don't know how old you were, Sarah, but that she said that she was going to study dolphins and skin lesions one day, and here you go. Here's the nice culmination of that. So, I felt like it was just a nice thing to share with the Polgar team and everybody here, that this is something that Sarah's been very committed to for a long time. So thank you.
Well, not just dolphins, but specifically skin lesions. That's very specific.
There was actually online evidence of it as well. So yes, it was pretty remarkable. So you learn something new every day about your students. So.
We do have a couple more questions, and we're doing great on time. So if anyone else wants to ask, please continue to type them into the chat. The next one is from Gregory O'Mullen. He asks, do you think it is possible that the freshwater correlation may be an indication of more pollution sources in estuarine as opposed to open marine water?
Sorry, could you repeat that?
Sure. Is it possible that the freshwater correlation might be an indication of more pollution sources in that estuarine water as opposed to the ocean water having less pollutants coming in from the open marine sources?
I see. It could very well be. That's again, we just looked at salinity, weekly values for salinity, and I think future studies, like I said, should look at chemical pollutants as well in order to kind of tease that out a bit more. For example, whether there's mercury in these waters, things like that. So again, I think there's lots of room for further research on this population and what's causing these lesions and what they mean.
Great. And another question from Ellen Hartig. In 2020, were dolphins seen closer to the New York, New Jersey shorelines? Saw news coverage elsewhere that the Chinese white dolphins were spotted in Hong Kong with less boat traffic. If observed here, would less boat traffic due to the pandemic be part of the reason?
So as far as I know, there's been no evidence to suggest that. It's something we could look at. We didn't really look at how far from distance to shore in these sightings, and if those change significantly over time, and compared to earlier years. But it very well could be. We just haven't looked at that.
Yeah, good suggestion for some further investigation for you, maybe.
Yeah.
And Louis Kindman asks, did I miss seeing the population totals for each category?
Oh, I see. For the categories of skin lesions?
I think so.
I can go back. So this is the number of individuals in our catalog that had it out of 203 photographs of individuals that were used in the lesion analysis.
Great.
If that's what you're referring to, yeah.
Yeah, Louis, if that doesn't answer your question, just clarify that question in the chat if you don't mind. And that is the last question that I see, unless Sam, you're seeing something that I don't. Oh, Louis says that's fine. Thank you so much.
Thank you.
Okay, does any of the other panelists have any questions or comments? All right, Greg O'Mullen says nice job.
Thank you.
Oh, I see one more. Go back to that chat. I lost the chat. Oh, here we go. Ivan asked, do you know if Amazonian freshwater dolphins exhibit similar lesions?
I don't know off the top of my head. Yeah, I don't know off the top of my head.
Okay. All right. And a lot of comments from your fellow Polgar fellows. That was a nice presentation. So thank you so much, Sarah. And we will move on at this time to our next presenter. Emma Garrison is up next. She is a student at Queen College, CUNY. Her supervisor is Gregory O'Mullen, and she will be presenting on exploring the influence of urban sewage pollution on fish microbiomes in New York Harbor. Emma, I can see your presentation. It looks good.
Wonderful.
And I can hear you. Go for it whenever you're ready.
All right. Hi, everyone. My name is Emma Garrison. I'm a master's student at Queens College in the School of Earth and Environmental Sciences, as just mentioned. And I work with Dr. Greg O'Mullen. And my project for this fellowship attempted to explore the influence of urban sewage pollution on fish microbiomes in New York Harbor, as Sarah just mentioned. So, there we go. And so I just wanted to take a moment before I got started and acknowledge that this project would not have been possible without the assistance of this fellowship through HRF. At a time when a lot of projects haven't been able to come through to fruition because of COVID, it's amazing that we've been able to do this. And I also wanted to thank the New York Water Trail Association for some lovely citizen science-based data, as well as my advisor, Dr. O'Mullen, who spent many an hour working through this with me, so thank you. So my interests are broadly in how organisms can stand to survive in our urban environment. We're talking about dolphins coming back in the previous presentation, and I think that it's really amazing that we can see organisms living and thriving in our harbor. I grew up several blocks away from the Gowanus Canal, and we kind of thought, you put your hand in the water and it'll burn off. And that's, we don't want to be sticking our hands in the Gowanus Canal, but it's certainly hosting a wide variety of organisms that we wouldn't necessarily know. And so I've done a lot of work looking at helping with bioblitzes in the Gowanus Canal and doing other sort of education work looking at the organisms that live in this environment. Here I am documenting some critter with my partner, who is always willing to get into a canoe in some of the nastiest water. So it's really something that I love very much. And so my advisor, on the other hand, kind of looks at this urban environment from a microbiological perspective. And so this project attempts to sort of marry these two interests, looking sort of at the microbiological component of the organism in an urban impacted environment. And so what is this harbor urban impacted environment? And so this is a visual from Talley et al. That shows sort of water quality throughout the harbor based on a variety of parameters, including dissolved oxygen, nutrients, turbidity, things like this, giving them each a rating from, you know, D through B in terms of our harbor. And we can see that there's a variety of water qualities, I guess, throughout our harbor. Specifically, I want to draw your attention to the fact that places like Flushing Bay, Newtown Creek, the Gowanus Canal, where I'm in that canoe, have some of the worst water quality, and that these are highly localized conditions, and that all sites within the harbor are not created equal. If you're sampling in the lower bay, you're going to see much better water quality than somewhere like the Gowanus Canal or Flushing Creek. And so what are the sources of this contamination? Well, we have historical pollution that has contributed lots of contaminants from, you know, past industry. But we also have modern pollution in the form of our sewage system. So our sewage system was set up to take all of the waste from all the wastewater from our homes, as well as all the stuff that goes down the drain on the street in our storm sewers, and it combines that and it goes off to a wastewater treatment facility where it gets cleaned and processed. However, when it rains, the system is overwhelmed. And so instead of that backing up into our homes and our streets, it overflows directly into the harbor. And with it, it carries things like PAHs, heavy metals, debris, just general trash, as well as microorganisms, right? Remember that this is, you know, including human waste, as well as other organisms like dog waste from the street. And this environment that's incredibly impacted hosts a variety of species, not just that one that I was photographing in that canoe, but a wide variety of fish species that we really care about, that are on high priority lists for conservation, whether these be permanent residents of our harbors, like the sweet little mummy chog right here that were in that first video that I showed, or organisms like American eels, which are, you know, stopping through the harbor on their way up, further up the Hudson into, you know, into like more terrestrial environments, so to speak. And so whether it's a pit stop or whether it's a permanent home, fish are living in this environment. And in turn, this contamination will impact fish health. So fish are exposed to poor water quality through a variety of pathways. The best visual for, you know, kind of the drastic impact that contamination can have on these organisms are exhibited in giant fish kills that happen, you know, for bunker. And this video was taken in Flushing Bay from Hudson River Keeper. And so we see that these fish are being impacted. It's not just large fish kills that we still don't fully understand what's going on with, but it's other things like immunotoxicity and growth inhibition, abnormal mortality, lack of reproductive success. And so one potential mechanism that we are not exploring, right? We have a pretty good understanding of how these, you know, chemical contaminants impact these organisms, but we also can potentially see this through fish microbiome. So what is a microbiome? It's kind of an ecosystem on a microbial level. So it includes the microbiota, all of those microorganisms that live within that environment, as well as their genes, which is important because it's kind of like their potential, what they can potentially do in their environment. So it's not just, you know, the squirrel and its environment, it's also what the squirrel is doing in the environment, what the microbes are doing in the environment. And we have found recently through a lot of research that gut microbiomes play critical roles in fish health. And we know that, our understanding of the role of microbiomes in human health is more recently being better understood. But it's also being studied in fish, primarily in the realm of aquaculture. But we have found that, development, nutrition, fecundity, survival, all of these, pretty important things are often impacted by fish microbiomes, and a disturbance to that microbiome can really impact fish health. And so how does this really work in the context of New York Harbor? Well, there's many pathways for microbiome contaminant exposure. Like, we understand that a fish being submerged in pHs is going to experience, a lot of difficulty. But there's other potential pathways. The first thing is that early environments can shape microbiomes for fish. So those organisms that grow up, so to speak, in our harbor are experiencing sort of this microbial and chemical environment that is impacted by CSO, or combined sewage overflow. But we can also see that in this visual, fish can congregate around combined sewage outfalls. This visual shows here our caution wet weather discharge point, meaning that this is where that stormwater and combined sewage runoff enters our harbor. And we've got some schooling fish below that. And granted, this is not an actively flowing CSO. Unfortunately, those are really difficult to capture because of all of the turbidity and gunk coming out. But it makes sense that fish would want to spend some time here, right? When we see this is a CSO in Florida and off the coast of Miami, and fish congregate around these outfalls and actually feed directly at them. And this makes sense, right? Because there's, we're grossed out by CSOs. There's a wide variety of kind of tasty detrital food and microorganisms and other things that lower trophic levels can feed on. And then this can move up trophic levels as the small fish come in, larger fish come in, and then even larger fish come in. And it goes all the way up to birds, where you can see birds, in fact, congregating around combined sewage outfalls. Additionally, there's been some work that was done by my advisor in conjunction with HRF that looked at the fact that sediment can serve as a reservoir for the contamination and microbiota that come out of CSO outfalls. And fish are known to live a benthivorous foraging lifestyle, where they will go and sift through sediment and pick up invertebrates and eat those. And so there's kind of two potential pathways in terms of like direct sewage influx into the environment. But despite all of this, right, we understand, okay, the environment is impacted by CSO. We understand that fish microbiomes are impacted by, are playing critical roles in fish health, but understanding the context of urban pollution and fish microbiomes is still lacking. And so I set out to better understand the interaction between urban sewage pollution and fish health, specifically through the lens of their microbiomes. But typically what we use to understand CSO impact, how much volume of combined sewage is entering the harbor, is through a fecal indicator bacteria. Predominantly we use Enterococcus, which is bacteria that lives in the guts of warm-blooded animals. But if you're looking at guts, how do you use an indicator that comes from guts? It doesn't really work. And so we needed to find sort of a metric that we could use for CSO impact. And so we looked at previous work that was actually done by a Polgar fellow through this program, which is really neat that those were able to tie up, that looked at the relationship between fecal indicator bacteria and antibiotic resistant bacteria. And so you can see here that we have the top graph looks at ampicillin resistant bacteria, so a specific type of antibiotic, and the lower graph looks at tetracycline resistant bacteria, another type of antibiotic. These two antibiotics attack cells in different ways, and so when you use both, you get a better understanding of sort of the overall antibiotic resistance present. And then we can also see Enterococcus along the bottom. And what these tell us is that these antibiotic resistant bacteria and Enterococcus are well correlated, positively correlated. And so if we are seeing Enterococcus in our water, we are likely seeing antibiotic resistant bacteria as well. Furthermore, this study showed that during wet weather events, when combined sewage is actively being pumped into our harbor, our Enterococcus increases, our tetracycline resistance increases, and our ampicillin resistance increases. And so we know that CSOs are playing a pretty big role in delivering antibiotic-resistant bacteria into the harbor. So, can we use antibiotic-resistant bacteria to look at the patterns of persistent contamination, wet weather oriented, and is this reflected in fish microbiomes? And that's really what I set out to study. So we wanted to look at this contamination along a gradient. And so we can see these are the 3 sites that I explored. Obviously, we would like to have more sites, but we were limited because of COVID. But we can see here Flushing Creek, Little Neck Bay, and Hobart Beach. I should point more at the arrows. And while we can see that these are sort of a gradient of, sort of urbanization, right? We're going from Flushing Creek, which is in the middle of Brooklyn and Queens, out to Little Neck Bay, and you can just see this in the green levels, but also population density shifts drastically. We can also see this sort of gradient in terms of sewage infrastructure. And so here at Flushing Creek, we have one of the biggest combined sewage outfalls in New York Harbor. In Little Neck Bay, by comparison, it's a much smaller pipe that is primarily stormwater runoff that comes just directly from streets. And then Hobart Beach is, you can see here, it's sort of this spit that comes off. And so it's not receiving a ton of stormwater runoff directly from streets. And it's actually a really beautiful place. So we're going from, really big CSO to a really nice beach that was a lot of fun to spend some time at sampling this summer, or this fall. And just to kind of drive this point home, we can see sort of, these are the combined sewage outfall locations within the harbor, and the size and color indicate the volume of CSO that's discharged. So here's our Flushing Creek site relative to our Little Neck Bay site. It's very different. So if we look at historical data, just to kind of drive it home one more time, we can see this is based on data from the DOH, from the Department of Health, as well as the New York City Water Trail Association, that our fecal indicator bacteria do a good job of indicating that these sites are statistically different, and that Flushing Creek has the poorest water quality and the most CSO impact, and we have our gradient. Unfortunately, when we went to study this with our sites, we did not find this. And our fecal indicator bacteria sampling did not result in the same sort of gradient. These sites were not seen as significantly different. However, the likely culprit is undersampling, and so future work attempts to sort of increase our sampling size so that we can better see this. This is a well-established trend, even though our work did not directly reflect it at this time. And so for the getting into the microbiome sampling itself, we used cultivation-based enumeration to quantify and compare heterotrophic and antibiotic-resistant bacteria. So basically, we have Petri dishes that have media on them that can grow either all of the organisms, or we spike them with antibiotics, and so all the organisms that grow are antibiotic resistant because they're surviving in an antibiotic environment. And so we can use this to count individual colonies, which are related to single cells, in order to quantify the amount of antibiotic resistant bacteria, but we're not capturing all of the organisms, right? We're only capturing the ones that are growing in the specific environment that we've given them. And so we use sort of non-antibiotic resistant or non-antibiotic plates in order to compare heterotrophic microorganisms, because we want a relative measure, and that enables us to kind of use this method, even though we're not looking at every single microorganism. So we looked at these in the water, and we looked at them in the microbiomes of Fundulus heteroclitus, our mummy chogs, that we trapped using baited minnow traps, and we dissected and extracted the gut contents of. One major assumption that we did, in part because of a COVID precaution, was that we assumed that fish gut microbiomes are, fairly regulated. They are inside the fish. They're not directly reflecting the environment around them. because we didn't want to go out sampling during active sewage events. So mummy chugs as a model organism, why might we use them? Well, they have a really small home range of about 200 meters, and they are resident species in some of the most impacted waterways in New York Harbor. So you can find them in really clean water, but you can also find them in the Gowanus Canal, in Flushing Bay and Flushing Creek. That first visual that I showed you was from the Gowanus Canal. So they're living in all of the environments. Also, they're very cute. But anyway, so they're a really solid model organism. And when we compare the fecal indicator bacteria that are found in the water with the antibiotic resistant bacteria also found in the water, similar to what Suzanne Young's work did previously, we found similar statistically significant trends that fecal indicator bacteria and antibiotic resistant bacteria are again positively correlated. And then when we look in the guts of our mummy chogs, we found that 95% of individuals had detectable antibiotic resistant bacteria present in their guts. So we know that these environments are not mutually exclusive, that there is a level of interaction that is occurring between the environment and between the fish microbiomes. Additionally, we were able to find that, so these are the ampicillin resistant cells present in the gut versus the ampicillin resistant cells present in the water. And when we compare those across all of our sites, we can find, again, a positive correlation between the presence in the water and the presence in the gut. And this tells us a number of things. First of all, it means that our guts are being, the fish guts are being impacted by the surrounding aquatic environment. And we also know that this is reflected in the water. And finally, we can probably say that, if we know that fecal indicator bacteria and ampicillin resistance in the water are strongly correlated, then fecal indicator bacteria and ampicillin resistance in the guts, or antibiotic resistant in the guts, may also be positively correlated. And this needs to be further understood. So future steps. The first thing is we need more fish. We need more samples. We did not see that trend in just a general fecal indicator bacteria for our sites. And so if we have more samples, we are more likely to get a better understanding of our trends and be able to see sort of if there is a difference between sites, potentially looking at wet weather versus dry weather events as well. And we also want to use molecular approaches that look sort of holistically at the DNA, right? Our sampling method was only able to select a small sampling of our microbiological organisms, and so molecular approaches could let us look at everything that's going on. And we can also sort of take samples of those individual colonies and use that isolate information to characterize, who are these microbiota that we're finding in the water versus the gut? Are they the same organisms or are they different? So finally, I wanted to give another thank you to the Hudson River Foundation for supporting this work under such difficult circumstances. I also wanted to thank the audience. Thank you for listening to my project that's gonna play a pretty big role in my thesis and for supporting this. So thanks again, and I'll take any questions.
Great, Emma, wonderful job. And again, if you have any questions or comments, please type them into the chat and I will field them for Emma. So you have a few already. Louis Kleinman asks, this is from my curiosity, why did you not use the Riverkeeper data in addition to water trails?
It was just based on our sites, specifically where we had located some data from. We wanted to get data points from right next to where our sites were, and that's where the water trail sites happen to be.
Great.
Basically, just one more thing, sorry, because we know that things are so localized that having data that comes almost directly from where you're sampling your fish, you're more likely to see that relationship.
Right. So another question from Tim Gilman. We have a floating classroom in the Gowanus Bay. If you're interested in coming to speak with school kids and visiting us, you can get in touch with him.
Oh, that's great.
got his-
I spent a lot of time working as an environmental educator at the Gowanus Canal Conservancy and doing work with the Mary A. Whalen at Portside, New York. So I've done a lot of educational events. Happy to talk to students. Yeah.
Tim's email is in the chat. We'll get that to you if you'd like to get in touch with him. Rachel Davis said that she had to run, but it was an excellent presentation. She wanted to share that we have an upcoming workshop for students and anyone new to advocacy seeking tools, ideas to build resources, protection, and innovative solutions through community engagement. I assume that's for any of our Polgar fellows, and she includes a website link in the chat as well. Georgia Coleman asks, well, first says, cool study. Do you have a minimum size of mummy chug to do the study from?
Yeah, so well, there's kind of two issues here. If we get really large one, we run the risk of sampling a different species of minnow. And so we kind of kept it to fairly small ones, but I would say that they were, usually around 3 inches.
Great. Again, we've got plenty of time, so feel free to keep typing questions into the chat. We have another question from Dorothy Petite. Do you think the marsh area in each region, marshes help keep the water clean, might be correlative with the bacteria?
So marshes, yes, help keep things clean, but they also complicate things because especially with, and that's not to say that we don't love marshes, but we do know that lots of birds and other organisms are going to be spending time in marshes. And so that can also increase fecal indicator bacteria in the water, and it might not be sewage related. And so that actually sort of complicates things more than for this particular study, obviously, than helping things in a lot of ways. So we kind of distinctly chose these locations that were not immediately adjacent to, marshland. Yes, they're probably providing a larger habitat. They're probably actively cleaning the water. But again, these are really localized conditions, and we're looking more at this sort of, sewage impact that is more immediate.
Right. Brendan Garrison asks, are microbiome abnormalities passed from prey to predator?
That is a really interesting question. So that is something that I'm really interested in studying. That's hilarious. This is my brother, and
Garrison.
I know to ask this, and I did not tell him to ask this, which is hilarious. But I think that would be really cool. I think that, looking at, if there's any trophic level implications, obviously, because we're seeing at these CSO outfalls that, we have many trophic levels that are interacting with CSOs. If you can, see how this moves. Unfortunately, the methods behind this is such an understudied thing that it's really, we're kind of just getting the data on this, but I would love for future work to involve looking at up trophic levels.
All right, I don't see any more questions in the chat right now. Any more questions or comments from our panelists?
I have one. It's really meaningful work. Do you know if, off the top of your head, like what any solutions to CSOs there are in the city?
Yeah, there's a lot of solutions that people are working on, using things like wetlands and oysters to actively clean the harbor are some. There's kind of, can we use green infrastructure on streets to absorb some of that stormwater before it enters into our sewage system to kind of control for wet weather events. We can also, build really large holding tanks that will hold on to sewage and then slowly deliver it to our wastewater treatment facilities after the storm event when their capacity basically is increased, or, they're not as stressed. But again, these solutions, green infrastructure can help in a lot of ways. Big storage tanks can be complicated because, they have to be built underground and people live in this city. It's really densely populated area. So it's difficult to, find really good locations to do that sort of thing. And there's lots of complications that come with that. And additionally, these aren't always, these are projects that are going to take really long periods of time. And so, yes, actively being worked on. Yes, many solutions that we're actively pursuing, but immediate results, probably not.
Okay, we do have some more questions popping up in the chat. Another one from Lois Kleinman. Is it possible that fish congregate around CSO outlets are, in addition to the nutrients, are also influenced by changes in water temperature?
So I'm a little bit confused, but I'm gonna run with it and hopefully I answer this question appropriately.
Okay.
So yes, CSOs are delivering kind of what some organisms consider tasty, delicious things. Temperature plays a role maybe in that, we have more CSO events that occur during summer and autumn months due to, weather patterns. I think it would be very difficult to tease apart sort of the role that temperature plays in this, even though, its overall temperature might be representative of, larger scale changes and shifts in the environment.
Okay, so Louis, if that answers your question, let us know. But Louis also wanted to answer the other CSO question about the cost of mitigating that would be a large amount of money in the billions. And thanks you for your answer to his other question. I'm seeing lots of nice jobs on the chat, lots of thank yous for your answers. And unless anything else pops up or any of the other panelists have questions or comments, I think we could move on, and Dave Yazo will be facilitating the rest of the presentations.
Okay, thank you, Sarah. So our next presenter is Aslan Makpool from Queens College, CUNY. His research is entitled Investigating the Distribution of Legionella in a Freshwater Hudson River Watershed, and his advisors are Greg O'Mullen and Eli Duker.
All right, thank you for that introduction. Hi, everyone. As you said, my name is Aslan Makpool from Queens College, and my project is on investigating the distribution of Legionella in urban and suburban Hudson River watersheds. So, how did I get to this project? What led me to study Legionella? Well, it started in my undergrad at Bard College, where I met my advisor there, Eli Duker, who was studying kind of the microbial interactions between air and water interfaces. So you see on the right-hand side, that's kind of a figure from some of his studies dealing with bioaerosols and such. And then this paper in my senior year I read and was kind of instrumental in going into the work I did for my senior thesis. So this paper was titled "The Local Environmental Pollution Strongly Influences Culturable Bacterial Aerosols at an Urban Aquatic Superfund Site," which was Newtown Creek. And so this got me thinking in terms of bioaerosols and kind of their effect in urban centers. So this led me to my thesis. So a bit of the background, metadata from a related study in Newtown Creek. My advisor alerted me that Legionella was detected in Newtown Creek, and which we knew was a bacteria of concern, especially in New York City. So that kind of combined what we knew about Legionella with kind of the work he's done with bioaerosols. So my senior thesis work was kind of starting to get at that, answer that question as to whether we can detect it in air or not. But most of it was focused on this device called air impactor. That kind of got me into Legionella. And then after I graduated Bard College, I moved to Queens College, where I met my now current advisor Greg O'Mullen, and this kind of broadened my scope of Legionella and kind of looking at it at a wider environmental scale. Okay, so what is Legionella? Legionella and why Legionella pneumophila? So Legionella is a gram-negative bacteria and became known after this its identification at in 1976 American Legion conference. Following the conference, there's a wide outbreak of participants, and no one knew why, what was going on, until the CDC, I think later that year, a year later, identified Legionella as the culprit behind that outbreak. So as of today, there's around 58 Legionella species that are described in literature, and of those, 25 are linked to disease. Legionella pneumophila, though, is the species which causes majority of the infections that we see today. Okay, so most people, when they think of Legionella, they think in terms of an airborne pathogen, mainly linked to kind of cooling towers and such. But Legionella is known and reported to have a widespread environmental distributions. Several studies have found it already in soil, lakes and river systems, as well as man-made water systems. So aside from cooling towers, you see it in sprinkling systems, hot water tanks, and such. So it is known to have a widespread occurrence. So in terms of New York City, Legionnaires, so I want to talk a little bit about the relevance of wastewater treatment plants. So wastewater treatment plants, we know, are growing in the number as urban centers grow and populations grow. In the literature, there has also been connections with the conditions created in wastewater treatment plants, which have been conducive to proliferation of Legionella in different stages. The temperature above 37 Celsius is ideal for Legionella growth. Also, the addition of biological aeration tanks, things like that. There's also been studies in a particular 2018 study found that these facility also can play important roles in community and local transmissions of Legionnaire's disease. So coming to Queens College, where kind of in Dr. Mullen's lab, he kind of studies the distribution of sewage-related microbes. I kind of wanted to see if Legionella had that similar pattern of sewage-impacted waterways and the kind of microbes we find in those waterways. Okay, so this is what led me to my initial study in the summer of 2019, where where we studied Legionella kind of layered on top of the sampling that we do for Riverkeeper, and there's 22 sampling sites that we checked for Legionella, and 15 initially were estuarine sample sites. This was over a four-month period, and on the left-hand side, you can see it's primarily the East River and Western Long Island Sound was the location of the initial study. And we at these sites, we measured dissolved oxygen, temperature, and salinity. And then back in the laboratory, we ran Legilert, which is an IDEX assay to identify Legionella pneumophila, specifically Legionella pneumophila, and then also Enterolert, which is a common fecal indicating bacteria that we study in the Oman laboratory. We also did Stervex filtered the samples for DNA analysis. Okay, so with this initial data, we saw that there was a positive correlation between fecal indicating bacteria, so Enterococcus and Legionella, but it was a very weak sort of correlation in the estuarine environment. We did detect Legionella pneumophila in 51% of the samples, but that the bacteria was found mainly in low levels. So we also compared it in wet versus dry events. As commonly seen with fecal indicating bacteria, wet weather events tend to have higher concentrations, which Legionella kind of emulated in that sense. So we did see a significantly more concentrated Legionella in wet weather events. So looking kind of beyond what we've saw in the estuarine environment, we wanted to see if there we could see it elsewhere besides just the estuarine environment, and this is kind of what led to sampling stormwater, and this also is from the previous work of Angel Montero, who was a previous Polgar fellow who sampled urban street water for fecal indicating bacteria as a source to waterways. So what Angel found was that fecal indicating bacteria were abundant in all of the street water samples that he sampled, and the concentration was significant enough to indicate that it is a source to waterways. So that was kind of a big motivation in terms of me also going into sampling street water to see if Legionella was there as well. So this is where I layered street water samples on top of the estuarine sites that we already had. So we sampled 5 different street water sites, and we did find, in fact, a correlation between Enterococcus on the x-axis. You have Legionella on the y-axis per 100 mil. And we saw a correlation between those two when looking at street water, and it was significant. So, so on top of this, we also looked at 2 CSOs, as we know that CSOs are a major contributor to of sewage to waterways. And what we found here was that CSO and street water were not significantly different. So therefore, it can indicating that street water can be a source to waterways. And these, again, on top of this, these 2 sources are higher than the estuarine values that we saw for Legionella. Side note, there was the max value of a Legionella was found it was found in an estuarine site, but this site was very close to BB08, which is one of New York City's largest CSO pipes. So the summary of this New York City project that we did in the summer of 2019 was that the results indicate that there is a strong connection between wet weather contamination from street water to the estuarine environment. A side note, we did confirm the detection of Legionella pneumophila as well as we serotype strains 1, 5, and 6, which are the strains commonly associated with infection, and this was from independent laboratory EMLS. So this brought me into the current study that we did upstate in the Hudson River estuary. And so the significance of it is that the data would provide us a unique contribution to the larger literature on Legionella ecology. It also we also wanted to see if, since the preliminary data indicated that stormwater was a major source to the waterways, we wanted to see if that was more broadly applicable to the upper Hudson River estuary. Okay, so these are the initial objectives that we had for this project. It was kind of more broad in scope, but then obviously COVID-19 hit and kind of uprooted that. So we had to rehash and like refocus the project and focus on kind of the main points of what we wanted to see. So yeah, so in fall of 2020, we started sampling. There was a total of 12 sites that we regularly sampled from September to November. 4 of those were estuarine sites, 5 were street water sites. So in the lab, again, we used a lead alert to culture the samples and to identify Legionella pneumophila. And then in parallel, so on the left-hand side, you can see there's an image of Grace Carter, who works in the Duker Laboratory upstate at Bard, who helped tremendously in the sampling effort, as well as parallel analysis of some of these samples. So she ran up there in the Duker Laboratory enterolert, coliform, E. coli assays, as well as collecting DNA from some of these samples. Also at the bottom, you can see an example of the trays of Legionella and what constitutes as a positive detection. So the main objective in this sampling of in fall 2020 was to determine if Legionella pneumophila is present in suburban street water and also the freshwater Hudson River waterways, which are much lower density in comparison to the urban center of New York City. So could we detect it? Yes, we did in fact detect Legionella pneumophila. It was in 22% of the estuarine samples. Again, though, in much lower concentrations in the estuarine samples. But in comparison, we also detected it much higher concentrations in 88% of the street water samples. On the right-hand side, you can see at the top where my cursor is, these are some of these street water sites that we collected. This is in Red Hook, New York. It's a small town adjacent to Bard College. And then at the bottom, you see sites for the estuarine sites that we collected, which were in Kingston, New York. So yeah, again, we did detect it. So this is a scatter plot correlating Enterococcus and Legionella. And it wasn't as significantly correlated as we saw in the urban environment. Then looking at waterways, we saw that street water though was much more concentrated than the estuarine waterways. As you can see, a significant difference there. So looking then against the New York City data and the suburban street water data, there wasn't a significant difference in this in the data and the concentrations of Legionella that we found, which is a good. I mean, it's a good thing in terms of what we expected to see, since we saw that street water was higher elevation and therefore a source to waterways. We saw a similar thing in the suburban street water upstate, where the concentrations were relatively the same. The fine line that I have right here is sort of where the detection limit was below. So that is just a side note there, but the ranges were in a similar concentrations that we believe that that we would see. All right. So conclusions and next steps. So these findings of the investigations did reveal a widespread occurrence of Legionella and included places that we didn't previously think would harbor the bacteria, that being street water. There is still more data to come, so ongoing molecular work is still happening, and confirmation of the Legionella pneumophila that we detected up in the Hudson River, upper Hudson River estuary. We're also looking at now soil and sand samples to see if that could act as a potential source to these street waters samples that we detected. And I just want to mention here that it is not a cause for panic that we detected Legionella where we found it. I want to remind you that we did that there are other confirmed sources, that being soils, lakes, cooling towers, wastewater treatment plants, and aerated waterways. What this data does reinforce, though, is stormwater is of microbial concern, and it kind of reinforces also the management of those aerosolization sources. Okay, and last but not least, I want to acknowledge a big thank you to my advisor, Greg, my previous BARD advisor, Eli Duker, in his laboratory, also Grace Carter, who's a BARD College senior, who helped tremendously in this fall sampling effort, Queens College, and then of course, big thank you to Hudson River Foundation for funding this project. Thank you.
Okay, thank you very much, Aslan. Great job. We definitely have time for questions. We are running ahead of schedule, so if I don't see any in the chat, but if there are any questions among the panel, feel free to chime in. In the meantime, I do want to make a comment, Aslan. I'm glad to see that you brought up the fact that you had to be adaptive to the COVID situation in your research schedule. That's something we had to really think about in continuing the program for this year and in selecting the Polgar Fellows. We wanted to make sure that they were projects that showed that they would be adaptive and that they could be conducted under this scenario. And it's something we got to think about moving forward for 2021 as well, unless something changes drastically over the next several weeks. So I'm really glad you brought that up, and it is a major point that these projects have to be adaptable under these circumstances. Do we have any comments from any other presenters or any participants at this time? Let's see. Yep, we have some questions coming in. So here's a question from Dorothy. Do you think Legionella was here in past centuries? Would it be possible to find it in soils?
Yeah, yes, I definitely think it was present here in past centuries. The literature already points to that, like it's found in soils, it's found in waterways. It has this already widespread distribution that we've seen. And yeah, that's also the ongoing work I'm actually doing now. I'm trying to see if I can detect it in soil samples.
That's a great segue to the next question from Greg, which says, what would you like the next step in this project to be?
Definitely, I think the next step is to kind of go beyond the street water and see if there are like sources, that being soil samples and such. I also kind of want to look into the molecular data a bit and see if beyond Legionella pneumophila, if there are other Legionella species that we see more in some areas than others, that being street water samples, and kind of seeing the wider distribution of that. Yeah.
Okay, well, thanks very much, Aslan. Unless we have any other questions, I think we're going to move on to our next, one more comment just popped in, a couple more. Okay, hold on, you're still on the hook, Aslan. Greg followed up and said, do you see any parallels to SARS COVID in terms of presence on surfaces versus in air, and which do we need to be most concerned about?
So definitely the airborne. So in terms of disease and stuff like similar to SARS-CoV-19, the Legionella is only kind of is only harmful in airborne form. And so I think a focus on those aerosolization sources rather than the surfaces is much more warranted than surfaces. I mean, also these surfaces aren't commonly aerosol like street water, for example.
Right. Okay, and then a comment from Lewis is that New York City has started to monitor COVID-19 in water treatment plants as a way of seeing changes in infection in the population. So that's a very interesting anecdote there. Okay, and yes, Ava says great job. we all think you did a great job. Everyone's done a great job, and I think we're going to turn it over to Eva now, and her presentation is entitled Contribution of the Hudson River Striped Bass Population to the Mixed Coastal Recreational Harvest at Montauk Point, New York. Eva Hasegawa from NYU, and her supervisor is Ike Worgan.
Hi, thank you for that introduction. So hi everyone, my name is Eva Hasegawa and I'm from the New York University School of Medicine. My supervisor is Dr. Isaac Worgan. And I'm going to be talking to you about the contribution of Hudson River striped bass population to the mixed coastal recreational stock harvest at Montauk Point, New York. So these are the fish that I'm talking about. They are anadromous and they spawn in major estuaries from Florida to the Gulf of St. Lawrence. And they tend to move north in the summer and south in the winter. So even though they're found from Florida to Canada, the populations in the center of its distribution are highly migratory along the Atlantic coast from North Carolina to Maine. And here, the populations that tend to be found are from the Hudson River, Delaware River, Chesapeake Bay, and the Roanoke River. So they're tertiary consumers. They feed on smaller fish and invertebrates and can get very large, as you can see here. They support a seasonal recreational fishery and charter fishing operations. So they're a glamour species of the Hudson area. So they support these recreational fisheries and charter fishing operations in the Hudson River and along the coastal waters, coastal Atlantic waters. So they're very economically important. 7.8 billion was given towards the U.S. gross domestic product. And that was the mostly recreational fisheries. So they accounted for 98% of the total. And in New York State alone, 1.165 billion was added to the gross GDP by the recreational fishery in 2016. So they've had kind of a tumultuous past in New York. So historically, the stock collapsed in the 1970s due to overfishing and habitat degradation. Specifically in the Hudson River, the commercial fishery closed in 1976 due to elevated PCB levels. So it just became unsafe to eat them. And to restore the stock, there's a coast-wide harvest moratorium that was imposed, and that did successfully restore the stocks in the early 1990s. And in fact, Maryland and Delaware imposed total moratoriums from 1985 to 1989. And there is also an amendment imposed to protect the 1982 year class of striped bass. And now, however, the Atlantic States Marine Fisheries Commission, the ASMFC, declared the stock overfished and overfishing is occurring. So they implemented harvest cuts in 2020. And as you can see in this chart, on the x-axis we have the year and on the y we have the striped bass per haul. So in 2019, there not many striped bass caught, young of the year striped bass. So it's below the 25th percentile. And so my study aims to determine the contribution of Hudson River striped bass population to the Montauk fishery in 2019. And by doing this, we can inform fisheries managers on the presence of major contributors that are reproductively isolated and that may vary in growth, maturation, mortality rates, recruitment success, migratory behavior, and exploitation. And we predict that we'll find a smaller contribution of the Hudson River population at Montauk in 2019 than observed by a previous study done by Bergman and Lieberman in 1978. This is because previously I showed that in 2019 there weren't many striped bass caught. And second, the proportional contributions to this migratory stock have changed over time based on their relative recruitment success in each contributing population. So we sampled locations, 11 different collection sites. But first, in 2019 at Montauk Point over here, 410 adult striped bass were collected by angling from June 23rd to October 31st. And these were provided by Captain Bert Prince of the Montauk charter boat, the Susie E. And we used fin clips as a source of DNA. So actually the captain's son took the fin clips and gave them over to us. So we were supposed to receive reference specimens from 2020 from the Hudson River, Roanoke River, Delaware River, and the major tributaries of the Chesapeake Bay. But because of the COVID-19 lockdown, we had to use past samples, so DNA from past years from these reference populations, and that actually worked out pretty well. So within the Chesapeake Bay, we have collection sites from the Patuxent, Potomac, Rappahannock, Choptank, Nanticoke, Pocomoke, and Upper Bay areas. And we have these collection sites as baseline populations to compare our Montauk specimens with. So first of all, we had to isolate the DNA from the fin clips. And we did a standard protocol with proteinase K, phenol, chloroform extractions, and alcohol precipitations. And we read the DNA concentration with a spectrophotometer. And then we did polymerase chain reaction, PCR. So we use microsatellites for this study, and microsatellites are repetitive DNA sequences that are non-coding, and there are several base pairs in length. So we use these 8 established microsatellite loci that have been successful in past studies with striped bass. And we also identified 4 new, we used 4 new primers or loci that were identified by this study here. So these were new to this, to the study. So then after we do the PCR, we have to measure or record the genotypes. So we do this by using a DNA sequencer to measure the PCR fragment lengths. So what it gives us is here the dye signal on the y-axis and the size on the x. So each peak represents an allele. So each fragment length corresponds to an allele of that locus. So in order to make sense of our data, we use a lot of statistical programs. First, we use something called GenePop, which measures the departures from Hardy-Weinberg equilibrium and linkage disequilibrium. So Hardy-Weinberg equilibrium is the genetic variation in a population will remain constant from one generation to the next, and things that can cause departure from this equilibrium could be mutation, non-random mating, inbreeding, genetic drift, things like that. And then we used Genodive, another program, to find out how different each fish from different populations are. So we used pairwise differentiation between populations. And then we used, in a similar way to do that, we used another program called Structure. But this program clusters the populations. It finds out how many clusters there are in the data set. So then we can use a program called Encore that does assignment accuracy. So it can tell how accurately the loci are in assigning fish back to their collection site. So it's sort of a way to make sure that we can be confident in our findings once we do mixed stock analysis and individual based assignments. So in Encore, we can also do mixed stock analysis to find out the relative contributions to the migratory stock. And lastly, we use gene class to do something similar, but here each individual fish is assigned back to a baseline population. So our first set of results are our loci. So we want to make sure that we have enough alleles to distinguish population, and it turns out there are a lot of alleles per locus. So the effective number of alleles are just the number of alleles with the same frequencies, and an FST is a measure of genetic differences among populations. So there aren't many major differences among populations in these loci. And all of these were in Hardy-Weinberg equilibrium, except for SB117D, which showed a little bit of departure, but not in most of the populations. So we kept it. So then we looked at pairwise differentiation among populations. So these are all p-values, and down here vertically under p-values, these are all collections, and horizontally is the same thing. They're all different collections. So for instance, in red, we have the Hudson River samples, and Hudson River fish are different from everything else. So our Roanoke, they're different from everything else, that they're genetically distinct. And Chesapeake, however, a lot of the tributaries can all just be lumped together because there are no significant differences between those tributaries. There are a few exceptions, but not many. They can all be grouped into one group. There were a couple though, like with Chop Tank and Nanticoke, they showed slight differences compared to other tributaries, but not much. And these are all using 12 loci, so all the loci we have available. And here as well, for our structure analysis, we also used the 12 loci to do these analyses. So here we found that among 11 sampling collections, we found 3 major populations, the Hudson, Chesapeake, and Roanoke. And Delaware can actually be lumped in with Chesapeake because they aren't genetically separate from the Chesapeake samples. And then we looked at just Chesapeake data, and these Upper Bay York, these are all showing the tributaries. And Structure actually found 2 distinct clusters in Chesapeake collections, and we're thinking that it could be Chop Tank and Nanticoke that are in one cluster. And this finding has been found in 2 other studies, and I'll get back to that. So here, this is assignment accuracy back to the collection site. So here we used a program called Encore, and we used a kind of analysis called 100% simulation. And this is where a fishery sample is simulated, where all the specimens are from the same population, and it measures how accurately specimens are assigned back to their sampling area. So this indicates how useful the loci are in assigning specimens back to baseline populations. So we want to make sure these results are fine before we go on with the Montauk specimens. And as you can see, when we use the 12 loci, assignment accuracy is pretty high. We have like 93% for Hudson, 90% and about 81% for Roanoke. So now that we have that settled, we can go on to the Montauk fish. And so why would we need to know their population contributions? Why is this important? So it's because spawning success and recruitment of each of these populations they vary over time, and usually they're asynchronous. So each population could require differing levels of protection over time. So that means we need a quantitative estimate of their contributions for effective management. And these population origin analyses are really important in determining the impact of mortality in coastal fisheries. So how vulnerable each population is to harvest. And for instance, we can do this specifically for Hudson River striped bass. So here is the first way we looked for population contributions through mixed stock analysis in Encore. And because I haven't finished genotyping for all 12 loci, I used 8 loci that I finished genotyping. So these two, mixed stock analysis and the next analysis I'll show is done with, are done with 8 loci. So here we found that the main contributor are the Chesapeake fish with about 88%. Then to a much lesser extent, the Hudson with about 9% and Roanoke with about 4%. So this was similar results were seen with another method we did in gene class called individual-based assignment. And here, each individual is assigned back to a reference or baseline population. And it gives like a confidence score as well, and how confident you can be that it came from that population. So here it gives us similar results to the mixed stock analysis. So 78% Chesapeake, 13.3% Hudson, and 8.8% Roanoke. So these results are actually comparable to what Bergman and Lieberman got in 1978. And Bergman and Lieberman didn't have the genetic technology to look at DNA, so they had to look at morphological characteristics such as, number of pectoral rays, fork length, head length, that sort of thing. And they found that Chesapeake fish were the main contributors, then Hudson, and then Roanoke. So what we found were that indeed we can effectively distinguish Chesapeake, Hudson, and Roanoke fish from each other, and that the 4 new loci that we identified are good markers for population assignment. And that as we found in previous studies, the Delaware fish are not genetically distinct from the pooled or individual Chesapeake population. So they can effectively be managed in one population. And that the Chesapeake fish are the main contributors at Montauk. And within that Chesapeake population, there could be a slight difference between the western and the eastern tributaries of the bay. So the Nanticoke and Choptank have shown slight differences in their DNA compared to the other Chesapeake populations. So that could be a good future direction for research. And that finding was also found in a study using short nucleotide polymorphisms and another microsatellite study done by Wergen et al. Last year. So there's still a lot of work to do. I completed 149 Montauk specimen genotyping. I have 261 left. So I have to do 4 more loci for those. And some loci are harder to genotype than others. It's just how it goes. So I'm going to be working on that. And then I'll be doing the mixed stock analysis and individual based assignment again, once I have all of those genotypes recorded. And also we are going to compare the population of origin of Montauk specimens depending on their collection time and their size. So there could be differences depending on how early in the season they're migrating, and differences in population contribution by size of the specimen. So I'd really like to thank my excellent supervisor, Dr. Isaac Wergen, and in the lab, Lorraine Masita. She is wonderful and so patient in teaching me how to do all the experiments and how to read the genotypes, because it isn't easy. It requires getting used to. And Dr. Nirmal Roy, he helped me with data conversion and data processing, which can be a real headache to do if you don't know what you're doing. So he really helped me with that. And we really have to thank Captain Burt Prince and the crew of the Susie E2 at Montauk Point, because they caught the fish for us. And his son, Justin Prince, took the fins for us and sent them to us. So this was his summer job at the time. And of course, I'd really like to thank the Hudson River Foundation. I'm incredibly grateful to be part of this community, and they've been so supportive and helpful during a trying time. So I wanted to include this. This is from the East Hampton Star. This is Justin Prince, the son of the captain, and he took some of our used some of our data to do his own project and won first place among 7th graders at a science fair. So it's really good to know that our data and our findings are going beyond the lab and beyond academia and inspiring a younger generation of scientists. These are some citations, and thank you so much for listening, and I'll take any comments and questions.
All right, thank you very much, Eva. That was an excellent presentation. Again, I'm glad you mentioned that there were some challenges related to the COVID situation. These are great lessons learned for those who might be on the call interested in submitting applications for this year's Polgar Fellowship program, and also good lessons learned for us as administrators of the program as to what works and what doesn't work. So really glad you all are bringing that up. Just a quick comment before we get to the questions. My first job from college was the data manager for the striped bass tagging program at the Hudson River Foundation. This presentation really brought back a lot of interesting studies and literature that I read many years ago when I was, again, right out of school in my first job in science. So I appreciate that. In terms of the comments or questions coming in, Lewis posted the fish consumption advisories from New York State regarding striped bass in various sections of the Hudson among different population groups. In the chat, if you can't see the webinar chat, you can get this information on the New York DEC website. So if you're an angler in the Hudson or thinking about being an angler in the Hudson and you might be considering keeping the catch, yeah, I should become familiar with those consumption advisories.
Okay.
Yeah, and then Greg has a question. Do you think that the Delaware versus Chesapeake population overlap is due to resolution of method? And if so, what approaches might be able to tease that out? Or do you think this reflects nearly complete lack of genetic structure differing in these regions? And very nice job.
Thank you. So I think this result has been seen in previous work that Dr. Worgan has participated in, and other studies as well that we're not affiliated with, that there really are no real differences genetically between the Delaware and the Chesapeake. Of course, with more loci added and more genotypes analyzed, we'll get more resolution. Other than that, I'm not really sure how to find more a difference between those two populations. I'm not sure if it's due to resolution of method or not. Yeah.
Can I comment?
Of course, yeah.
Yeah, there's something called the Delaware Chesapeake Canal, which connects the Chesapeake Bay directly to the Delaware River. And there have been several studies back 2 decades ago that showed that there was transport of striped bass eggs between the two systems. So that probably is the major reason why there are no genetic differences between them. Also, it's good to note, interesting to note that the Delaware population was probably extirpated back in the 1950s because of the oxygen block on the Delaware River around the Philadelphia area. So it was probably almost totally extirpated. So the stock has been reestablished starting probably in the 1970s and 80s. So that's another possible reason why there are no differences between the two.
Yeah.
Okay. And then, and so Greg follows up and says, So that would have differed historically then. Greg, you know, you can talk, right? You're actually a moderator here. So if you want to.
Yeah, sure. First of all, I thought it was I might as well go video as well then, right?
Okay.
I thought it was a great job, really interesting, and I think that also. So so that canal, when roughly would that have been built? And so historically, you think those would have been separated populations, but then the extirpation is a really interesting layer.
I don't know. I don't know. Possibly, but we really don't know. And I don't remember when the canal was built, but it was a long time ago.
Yeah, but that's a really interesting example of a potential, right, alteration in gene flow because of a physical structure like that. Really interesting, kind of the reverse of the dams, right? So interesting. Well done. I enjoyed the presentation.
Thank you. Thank you so much.
Okay. Do we have any other questions? If not, I'm going to turn it over to Helena for some concluding remarks.
Well, thank you very much. Wonderful presentations, everyone, and congratulations on a job very well done. I would like to point out that we do have Polgar advisors here today, and I see Ike just hid, but we have Dr. Gregory O'Mullen, Dr. Isaac Wergen, and earlier you met Dr. Howard Rosenbaum. I don't know, do you have any other comments, Greg or Ike, before we move on?
I would say I just want to comment that the C&D canal was constructed in the 1820s. Pretty old structure.
No, just a thank you to Polgar, right? This is really great for the students. It's also really great for advisors who have students who you want them to make progress during this period. So thanks so much for the extra work it took to pull this off, and you know, we appreciate the support, and as well as, you know, a real nice friendly, confidence building environment in which these presentations can happen. It's really great. Happy to be a part of it.
Well, thank you very much. We really couldn't do the program without the advisors, so I have to give out a special heartfelt thanks to you and for attending today and to our other meetings. I also want to thank Dave and Sarah Fernald, for all of their hard work, because they put endless numbers of hours into the Polgar program. So thank you very much. And I also have to do a special shout out to the members of the Hudson River Foundation Board of Directors who are on with us today, and thank them for their support over the years. So if you'd like to know a little bit more about the Polgar Fellowship Program, you can visit our website, hudsonriver.org, very easy website to remember. And if there are no other comments from the panel or from our fellows, then
just thanks Sam for facilitating and overseeing the technical.
Yes.
There he is. Thank you, Sam.
Thanks for that, Dave. Thank you, Sam, and thank you, everyone. And I hope you have a wonderful afternoon. Take care.
Bye, everybody.
Bye.
Thank you.
Thank you.
Thanks.